So next topic in this is translation. It's anyway you have studied it many times in biochemistry, in physiology and even microbiology, so I don't think you will find it difficult. You know that we have seen so far is after transcription the messenger RNA is made, and on that basis the copies of different RNAs are made. Ribosomal RNA is the RNA that has got complex and that forms the ribosome. If needed, we can study ribosomal RNA separately again, but then another RNA is there, that is a transfer RNA. So we have seen that there is a processing of the messenger RNA. We call it as a post-transcriptional processing, and in which we have seen that the RNA has got three important processes that are done. The first one is 5' capping, the second one is splicing, that is removal of the introns, and the next one is the 3' this adenosyl group attachment, which are around 200-300. Now this has made the messenger RNA complete after all these processes, and now this RNA will migrate into cytoplasm. It will bind to the ribosomal units and ribosomes, particularly either ribosomes separately or the ribosomes which are attached to the endoplasmic reticulum, to what we call it as a rough endoplasmic reticulum, and they will do the job of synthesis of protein. The figures are nice when they are self-explanatory. Let us see this figure in more detail if possible, if it can be enlarged. Yes. So this is the enlarged one. So this one, that these are the ribosomes. We know ribosomes are present in two subunits, smaller subunit and larger subunit. Whenever the messenger RNA comes and with the message that protein has to be synthesized, at that time both these subunits will come together. They will form a dimer, and this dimer, on that this messenger RNA will be there. Now here three nucleotides, they are forming one particular codon, and this codon is coming in this process. Now let us see one by one how this is happening. So this is the big RNA which is there. It has come to this ribosomal units, and these are the sequences. Okay. Now suppose these are the three nucleotides they are giving for arginine. Then here it were three, that is they are for proline, then glycine, serine, etc. whatsoever, depending on that. These are the three nucleotides which are there, and they are binding with these ribosomal units, and accordingly one after another these amino acids are being attached. Okay. So this is how the sequence that takes place. Here this RNA transfer is done by the transfer RNA, because, and they will have sort of anticodon like structure. This is codon and anticodon like structure which will fit into these codons, and because of that this whatsoever structure it is fitting, accordingly they will select the amino acid, and this there. So it will start with the start codon or methionine, and then this one after another, we call it as elongation, that will start, and thus we can see that amino acid sequence it has there, which has started, suppose methionine, asparagine, leucine, serine. See this we have seen earlier, serine, and thus it is forming peptide bond. At the end, when there will be last one, that is a stop codon, at that time this will be stopped, and this amino acids, this protein structure will be then removed, that it is there, that is the polypeptide or amino acid chain complete one, that will be removed. Now if required, then this will be having if it has need to be processed, then it will go to endoplasmic reticulum, where it will be processed and it will be packaged in a proper manner. So like this, the sequence like this, the action will be there. So this messenger RNA is decoded, and accordingly the specific amino acid chain is attached to make a polypeptide. So what are these three processes that are taking place? These are initiation, elongation and termination. We have seen these. So initiation, because whenever there is a start codon, then it will start with that. The ribosome will assemble around the target messenger RNA. The first transfer RNA is attached to the start codon. Then one after another, the ribosomes will be there. This will be transferring amino acid to the transfer RNA corresponding to the next codon. And the ribosome that will be moving, that is the translocation to the next one. So here the ribosomes are moving, or they are hopping from one codon to another codon, and this codon to codon hopping will take place, and it will create an amino acid chain to what we call as elongation. And lastly, there will be termination. When the stop codon is reached, the ribosome will be released the polypeptide, and then this will be this polypeptide will be released. As I told you, this polypeptide may be further processed and compact into some vesicle or something. If it is in endoplasmic reticulum or not, then it will remain in the cell. The ribosome, only free ribosomes will synthesize it, and they will put that protein, they will process the protein. Okay. So this is how this takes place. This translation occurs in the cytosol, or it can occur across the membrane of the endoplasmic reticulum, as we have seen, and it is called as a co-translation translocation, wherein the entire ribosome or messenger RNA complex will bind to the outer membrane of the rough endoplasmic reticulum, and this protein which is formed, that will be vesicalized and secreted outside the cell, because it is meant for translocation. Okay. Now number of antibiotics we shall see during chemotherapy, that they are binding with these various subunits of ribosomes, and only thing is why they are comparatively selective, or why they are causing lesser toxicity, because there is a difference in human beings. We call it as a total 80S ribosome. Out of that, there are two subunits. The larger subunit is 60S and the smaller is 40S, while in bacterial we call it as a 70S ribosome, wherein there is a 40S and 30S. So these are the reasons why the antibiotics are comparatively more specific towards microorganism, because there is a difference in the subunits size and structures, and therefore they will be affecting this ribosome, and they will affect the protein synthesis. So this is how protein synthesis is important, and how this is used in bacterial infection. The most important thing is we already, or many times I have told you, that most important thing is formation of protein. If you can see very good, this one is animation is given over here. If you can see here, I don't know whether you can see the cursor or not, but very good animation is there in this site, that is Wikipedia site. See how this is happening. Actually here we are finding that the RNA is moving, but it is not so. This is happening that, okay, see how the protein is formed and how the amino acids are attached one after another. So it's very good, this one, what we can say, animation is there, which will help you to understand. Now the next thing in this is, what are the basic mechanism? It is addition of one amino acid at a time to the end of protein, and this operation is performed by the ribosome, which is made up of two subunits. Already I told you about small unit and large subunit. They come together, already I told you, and accordingly they will be making it a polypeptide product. So here in animation you can see it easily how they are, both are coming together, and how it is hopping, and how there is a chain elongation one after another. Now next in that is, each amino acid is added which is matched to the 3 nucleotide sequence in the messenger RNA to what we call as a codon, and for each such triplet possible the corresponding amino acid is accepted. So this is how it is there. The addition of amino acid occurs in the C terminus of the peptide, and thus translation is said to be amino to carboxyl directed. Okay, that means initiation takes place with the amino, and then all the attachment happens to the carboxyl moiety. You might have studied the protein or peptide structure in biochemistry. So there are two terminus, one is amine terminus, another is a carboxyl terminus. So translation is amine to carboxyl, means first amine will be attached to ribosome, and then to this or carboxyl, the amino group, each amino group will be attached to the carboxyl later. It carries the genetic information about the ribonucleotide sequence from the chromosome to the ribosomes, and accordingly this triplet will be there. These codons will be read, and accordingly specific amino acids will be attached. They will translate the code for specific sequence. There are tables are available which shows which kind of sequence are there, and what kind of codon will give rise to what kind of message. From that, as we have seen, it can be start codon, it can be stop codon, and it can be amino acids for that. Each those triplet that are code for the specific amino acid, this translated into this code into specific sequence of amino acids, and it contains rRNA, that is ribosomal RNA, and proteins, and it is the factory. The ribosome is a factory where amino acids are assembled into protein. These transfer RNA are the small non-coding RNA chains, and they are having around 73 to 93 nucleotides in that, and they will transport amino acids to the ribosomes, and these have a site for amino acid attachment and site called an anticodon. So anticodon will sense it, and on that basis of anticodon the RNAs will be selected on that particular basis, and they will make it. Okay. The amino acid tRNA synthetases, these are the enzymes which catalyze the bonding between specific tRNAs and amino acids, and their anticodon sequence are called for. The product of this reaction is called as amino acyl transfer RNA, and this is carried to the ribosome by a particular transporter molecule that is called as EFTU, that is elongation factor. Okay, so this elongation factor is there where it is happening. These mRNA codons are matched through the complementary base pairing to the specific tRNA, and accordingly the activity will be there. Now amino acid RNA synthesis that are mispair the tRNAs. If this mistake happens, mispairing of tRNA with the wrong amino acid, it can produce mischarged amino acid tRNAs, and they can result into inappropriate amino acids in the respective position of the protein, to what we call as a mistranslation is important. Okay, this mistranslation here problem is not in genetics here. Okay, codons are correct, genes are correct, but the problem is occurring at this level, that it is not translating it properly. So this mistranslation of the genetic code naturally occurs at low levels in the most organism. Rarely it can occur because the system is so correct one. But in certain cellular environment, it can cause increase in the permissive mRNA decoding, and sometime may be for beneficial for the cell. One more topic related to this we are going to see later about the maybe next immediately, that is about various mutations, which already I am very sure you have studied mutation and all in biochemistry, but yes, still will make it once. There also we shall see what is the thing. The ribosome has three sites for the RNA binding. the amino acid sites are there, then peptidyl sites are there, and there are exit site are there, and accordingly these sites will be having one after another actions, and there will be bonding of the protein and formation of a protein. Okay, then the thing is, the next is, once it is there, once it is formed, then the translocation will occur. It will move the tRNA in a P site, and now it will be without amino acids, and then one to another, the charge with the polypeptide chain is moved to the P site, and accordingly these three important sites are there, that is A site, E site, P site. They will be working in a particularly sequence to make the protein formation. Okay. Now the next is various initiation factors are there into it, that this initiation will involve with the small subunit of the ribosome binding to the 5' end of messenger RNA, and it will cause the, with the initial factors, these initiation factors will important in the initiation of the protein synthesis. They will recognize the purine rich initiation sequence of the mRNA, that is called as a Shine-Delgarno sequence. This sequence binds to the complementary pyrimidine rich sequence in the 3' end of the I hope you are getting this, because here these are all purine rich. Purine means A and G, okay, that is adenine and guanosine, both are the purine, while cytosine and uracil, these are the pyrimidines. So in initiation factors, these are the purine rich initiation sequence on the messenger RNA, and the counteract to that is are the pyrimidine rich sequences are there on the 3' end of the ribosomal RNA. Okay. Now this complementary binding will be there. It will ensure that the 30th ribosomes is bound to the messenger RNA, and it is aligned in such a way that initiation codon is placed in the 30th portion of the P site, and accordingly one after another will be there. At the end, the stop codon will be inducing the binding or of a release factor protein, and this release factor, there are RAF, release factor 1 and release factor 2. They will cause the disassembly of the entire ribosome and mRNA complex by the hydrolysis of the polypeptide chain from the peptidyl transferase, which is center of the ribosome. The drugs or the special sequence motif on the mRNA can change the ribosomal structure, so that these tRNAs are bound to the stop codon instead of these release factors. So this translational read through is there, and it continues till the ribosome encounters the next stop codon. So simply the start codon will start and the stop codon will stop the translation. The process of translation is highly regulated in both type of organism, whether it is eukaryote or prokaryote. The regulation of translation can impact the global rate of protein synthesis. Global here, it doesn't mean that world. Here global means total. Okay, it impacts the total rate. Global means overall in cell. Okay, because there are global here. How we are using the one ribosome, another ribosome, third ribosome, 4 ribosome. There are many ribosomes and many copies of RNAs. So it doesn't mean that, or many ribosomes are working in tandem. So it doesn't mean that. So overall final rate of all ribosome, we'll call it as a global rate. So global rate of protein synthesis, which is closely coupled to the metabolic and proliferative state of a cell. In addition, the recent work has revealed that genetic differences and the subsequent expression like mRNAs, it can also impact the translation area in RNA specimen. Now where it is important, it is critical for the development and survival of the cancer. Okay, cancer cell must frequently regulate the translation phase of gene expression, though it is not fully understood why translation is targeted in the transcription. The cancer cell often have genetically altered translation factors, and it is more common for cancer cells to modify the level of existing translation factors. So many oncogenic, just now we have seen earlier the RAS pathway, then MAPK, then PIK3 or AKT, mTOR pathways. These all various pathways are there. We have not seen other pathways like WNTB, catenin, etc. We have not seen. We have seen only what our syllabus prescribed. So this we have seen, and in that we have seen that how these are working. Ultimately, reprogram the genome via translation. Cancer cells also control translation to adapt cellular stress. During stress, the cell translate mRNA that can mitigate the stress and promote survival. So these are the important things into it. These are the clinical significances of the translation. I will not go for this mathematical models of that. Only I like to add that there are basically 8 processes are there into it, and these mathematical models, they are predicting the rate of protein synthesis. So what are these? First is you need to have production of mRNA, that is final mRNA after splicing. Then second is initiation of this molecule with the help of initiation factors. So this is second stage. Then initiation of translation process, that is ribosomal subunits are recruited together. Then next is assembly of full ribosomes. Then elongation, that is addition of various amino acid into it. Lastly, termination of the translation, degradation of mRNA molecules, because whatsoever mRNA is there, it is degraded. That means now it will not make any copy of that. It is over. And lastly, degradation of a protein. So this is whole sequence is there, and for that each component has got its own time and its rate and volume. There are 3 important things: that at what time it will be starting, at what rate it will be starting, how many processing per given time per second or whatsoever, and what will be the global volume, as we have seen, what can be the global volume of it. So on that basis, these mathematical models are there. Particularly they are trying to relate with these changes with the various disease pattern or physiological pattern. So these are the mathematical models are there. For us important is these are the 8 important stages which are occurring in the translation. This 1 to 8 stages are there. Then genetic code, I think you can have, because there is nothing much to teach into it, that there are various codes are there species wise, and accordingly we call them that is these codes are referring for typical information. There are example, we have got start codon, we have got the end codon or stop codon, what we call, and then we have got various codes for different amino acids, and these are important. Now the very beautiful thing is there are many types of codons are giving same amino acids. Okay, there are many computer programs, they are capable of translating of DNA RNA sequences into protein sequence. Now this is performed using the standard genetic code, and some of the program can handle special cases also. So example, the rate alternative start codon for, that is CTG, will occur for methionine when used as a start codon, or leucine is there in other position. So this is how how these computer programs are working. Now these programs and this knowledge is helping again in one more thing is to make artificial synthesis, or to make synthesis artificially, and we have got various instruments for that. Okay, if we shall go in last one when we will go for biotechnology part of it, that we call we have got instrument like we call it as a PCR and RT-PCR, that is polymerase chain reactors and reverse transcription. Okay, that is reverse means RNA to DNA. So RT-PCR. So these are type of various instruments are available, and that are helping us to make various copies of these nucleotides and all. So there are, as I told you, there are a list of genetic codes and their meaning is there, and alternative codons are there. You can just, there is nothing to teach. These are just code AAA, AGU or like that. So whatsoever these 3 codon will cite for, this will be there, and accordingly these codes are there for. So they this much 33 different lists are there. These 33 are the list for different type of species. Standard, then vertebrate, mitochondrial one in yeast. What will be the mitochondria? We have seen that mitochondria has got a different set of RNA, and particularly it has got its own genetic material and own RNA, so that it will not depend on the nucleus, that it will do it and it will send it, because mitochondria has to function continuously. If you remember, we have studied this. So for that it will always have its own set, and accordingly it will be working. And therefore mitochondria has got its own code, the nucleus will have got its own code for whole other cell except mitochondria, and accordingly we can have several codes for different animals, different species, and these codes will be used. Fortunately, these are known, and accordingly it can be made. So here we finish what is translation. Then we shall go for the next will be various problems which are associated, or various changes which takes place to what we call as a mutation, and various reasons. So it's really a very clear idea now we have got. Now we are reaching to a very important information, that all the animals, all the species, for them there just like we have called cell as a functional and structural unit, similarly for that given cell the most important structural and functional portion is protein. And how protein is synthesized we know. Any mistake in protein synthesis can lead to defect either in structural or functional, and these structural and functional defects are nothing but the pathophysiology. And this pathophysiology, if we have to correct, now the scientists are trying to reach to this area, that can we correct the genes, because otherwise there is no reason it should happen. Okay, so we shall be studying the changes which are taking place, why the changes are taking place, and accordingly what can be done, what are the things which are responsible for various things. So this we shall be studying in the future course. If you have any difficulty, you may put it, you can ask, and I hope your studies are going well. So please keep your studies going on, and if you have any difficulty you can ask. Okay, take care and be safe.